Here we are in section 11.1. I've recommended that you watch this video rather than just using the book because the book kind of jumps around and I have some specific points I want to make. I think it's clearer in the video. The book does have some really good information, so I recommend reading the book too, but maybe take your notes from this video. The first objective is in chapter 11.1, and it says, explain the importance of the Human Microbiome Project. The Human Microbiome Project was a great big research project. It ran from about 2007 to 2016. It was funded by the U.S. government and with the help of some other governments and some private organizations. And what it did is scientists developed techniques before and during the Human Microbiome Project that allowed us to have a much better techniques at identifying the microorganisms associated with the body. And so what they wanted to do is they wanted to do a survey of the entire human body, inside and out, and find out what do the microbiota, what do the microorganisms associated with the human body in a normal, healthy human body look like. We didn't know, are they all the same in all people? Are they different? What parts of the body are there a lot? What parts are there a little? And we knew some of this, but with these new techniques, we were going to find out a lot more. What happened before the Human Microbiome Project is the way we identified microorganisms was we would take, we want to know what microorganisms are on your skin. We'd take a swab, we'd swab the skin, we'd put it on a petri dish and grow it up in the laboratory. We'd look for bacteria that grew. And so we analyzed our bacteria based on that, whatever grew in the laboratory. But what we were finding out prior to the Human Microbiome Project was maybe 30 to up to 50% of human bacteria were not culturable. And meaning that when we tested this bacteria on our skin, a lot of the bacteria that were there never even grew on that petri dish. And I think about it in terms of if, like an alien came to this planet and collected all different organisms. They collected zebras and oak trees and fish and otters and birds and all these different things. And they put them all in an aquarium to grow. Well, you can imagine that most of those would not do very well. And that's actually what we do with growing bacteria. We have a standard set of techniques we use to grow bacteria in the laboratory. And it turns out that that just doesn't work for a lot of microorganisms. So scientists started developing new lab techniques where instead of growing the bacteria, they actually looked for microbial genes. And so they did genetic analysis. So these are fancy laboratory techniques where instead of growing the bacteria, they looked for their genes. So now they did a swab and they did some fancy stuff and put it in a machine, and they identified the bacteria based on the presence of bacterial genes. The bacteria or the microorganisms didn't even have to grow. And what they found out is there were many more kinds of microorganisms associated with the body than we ever knew before. Now, we kind of understood this before, that the humans have about 10 times more microbial cells than human cells. And that number might be a little bit high, but there definitely are at least as many microbial cells, if not more microbial cells than human cells. And so what they did with the Human Microbiome Project, they got about 250 healthy people. They sampled them all over their bodies, and they did this genetic analysis to figure out what microorganisms are present in and on the human body. And they came up with this term microbiome. And what they meant by that were the microorganisms, their genomes, all their genes, and their environmental interactions. So the microbiome is a little bit more than just the bacteria. It's actually a little bit more complex. And when you hear this term, microbiome, you assume that the information that you're talking about was gathered using genetic analysis rather than just growing bacteria in the lab. So here's one of the parts, one of the reasons why I wanted you to watch this video instead of just read the book, because I think the book is really confusing with their terminology. And frankly, it's not just our textbook, but if you go on the internet, you can find different definitions for these words, and people use them in different ways. I'm trying to give you something that's pretty common and helps you understand what these are. And these are important terms. What we have is this term normal microbiota. Your book actually says normal biota, which I never use that term. You don't see, I don't know, it's not a very common term. Normal microbiota is a much more common term for what we're talking about. And what this means is these are the microorganisms naturally found in and on the human body. Okay, you test your skin, you're going to have microorganisms there on your hands, in your mouth, we're all over the place. And so these are the microorganisms that are naturally found in and on the body. So then after the Human Microbiome Project, or as that was going on, we came up with this word microbiome. And these are the microorganisms that are naturally found in and on the human body. Oh, the same thing as the normal microbiota. But these are identified by the presence of their genes. And so when I say normal microbiota, I'm just saying, I'm giving you a general term for the microbes in and on your body. When I say microbiome, I'm just being more specific, telling you that this was determined using these advanced genetic technique. So I'm telling you that we're finding all the microorganisms instead of just the ones that can grow. Your book uses these interchangeably, and a lot of people do. So if I say microbiome, you can think normal microbiota. If I say normal microbiota, you can think microbiome. Okay. The normal microbiota or the microbiome can have organisms that you can divide into two groups. We have the resident microbiota, and these are the ones that are typically present, living and growing in and on the body, on the host. They're tightly attached. So what can happen is here I do a swab on my skin and I grow up those microorganisms, and I'm going to see in that group, I'm going to see a lot of resident microbiota. Those are the ones that are growing, tightly attached to my cells. They're in between my cells. They're living on my body. They're multiplying on my body. The transient microbiota, as the word says, transient, they come and go. So just as I was able to grow some microbes from my skin that are residents, there's going to be some there that are there temporarily. I can touch the countertop. I can pick up some microbes. These microbes will be on my skin. They might be, they might be there as part of my normal microbiota for now. They were there when I tested this entire sample, but they're going to come and go based on, when I wash my hands or what I've been exposed to. So these are the ones that come and go easily. They're not actually living on my skin. Okay. I can think of an example like E. coli, which you've all heard of, is a common fecal bacterium. They don't live and grow on your hands. It doesn't mean they can't be on your hands. They can be a transient. And so, but once I wash my hands, they don't grow there very well. The environment's not right for them. So when I wash my hands, they generally are come off because they're more loosely attached. You're going to hear this vocabulary a lot this semester. I'll use normal microbiota, resident, and transient microbiota, and your book generally uses the word microbiome, but just think normal microbiota, microbiome, for our purposes, are essentially the same thing. So now that we have these techniques to identify microorganisms by their genes, what are some of the things that we're understanding now? In section 11.1, there's two different bulleted lists, but the first one has a whole summary of some things, and you might want to look at that. And I just took some highlights out of there. We have more microbial genes than human genes, and we are absolutely dependent upon those microbial genes for health. We just don't completely understand why or how right now. And there's a lot we don't know about what these microbial genes do for us. But we evolved in the presence of these genes, and the likelihood that they're doing very important things is very high. The other thing is, because viruses don't grow in the lab very easily, we didn't understand much about viruses being part of our normal microbiota. But because we could detect viral genes in our when we do study the microbiome, we know that viruses are a big part of our normal microbiota, and we'd like to understand more about what they're doing. Some normal microbiota are potential pathogens, and we kind of knew that before, and we're getting a better understanding of this. But it turns out that a lot of times we get sick. It could be from our very own normal microbiota. Now, most of them are preventing us from getting sick, but among them, there's occasionally a pathogen, a disease-causing microorganism that has the potential to make us sick. So some of our normal microbiota are potential pathogens. And then one of the huge things that just has limitless areas of study are our intestinal microbiota. That's where we have the most bacteria in our entire body, in our intestines. And we're finding out more of how they can influence our health. They play roles in obesity, causing Crohn's disease, diabetes, mental health. And it doesn't mean they cause these issues related to these, but they play some kind of role. Even autism has a role. People with autism tend to have differences in their gut or intestinal microbiota, and we're trying to understand more about that. The next learning outcome has to do with the steps that are required for a newborn to acquire their normal microbiota. And turns out in utero, in a woman's uterus, we used to think it was sterile, but now we know there's a small amount of microorganisms. Now, it's not a lot, but they do appear to be important, but it's not enough to colonize the developing fetus extensively. But there are some microorganisms that we might learn more about. And so what happens is, as the baby's being born, they're going to pick up microorganisms from the mother's birth canal. So their very first way of getting a lot of microorganisms is as they're being born. And this bacterium is going to come up a couple times this semester, this genus called Lactobacillus. You might have heard of Lactobacillus because it's in yogurt. You hear about it as a probiotic. And the women's vaginas have a lot of Lactobacillus. In fact, pregnant women have particularly large amounts. And this bacteria coats the baby as they're being born. The baby will continue to acquire more microorganisms and develop their normal microbiota as they have contacts with the environment, from touching people, from people kissing them, from putting things in their mouths, from putting their hands in their mouth. And the baby's going to gradually develop more and more microbiota. But in this early stages, where, shortly after birth, the baby doesn't have a lot of normal microbiota. And this is the reason why they'll tell you, don't bring your baby out in public. Don't expose them to a lot of things until they always say their immune system gets stronger. But their immune system's getting stronger because they're developing their normal microbiota. A complex microbiome is developed by late infancy. And in fact, there's certain things like honey, they tell you don't feed babies until they're 1 year old. And it's directly related to this development of their normal microbiota. And we'll talk about that later on. A lot of people are curious about C-section babies, and it turns out that C-section babies have somewhat different microbiomes than babies born vaginally. And what we do know is this difference lasts for at least 3 years, and the consequences of these differences are not well understood. You can imagine all the differences that a baby has, any two babies will have just in their environment. So trying to figure out what are the significant things about being born by C-section, it's a really difficult and long-term study you have to do to figure this out. So people are interested in this, though. And some people are even doing recommending that when a C-section baby's born, you take like gauze and wipe out the mother's vagina and then wipe that all over the baby to expose them to those microorganisms. In the next learning outcome, we want to identify the sites of the body where normal microbiota are found in humans. And so here's your highest number of normal microbiota are going to be the intestines, from the small intestine to the anus. So remember that stomach connects to the small intestine, that connects to the large intestine, and that goes out the rectum and the anus. And out of all this, the most bacteria are in this large intestine. So there's a lot of bacteria in all this, but the large intestine has the largest amount. The skin has a lot of normal microbiota, and the one kind of bacteria you're going to see a lot of is Staphylococcus epidermidis. When you think of Staphylococcus, think of skin, and epidermidis, you probably know that's a layer of the skin, the outer layer of the skin. And then the oral and the nasal cavities have a lot of bacteria. And particularly, you're going to see some Staphylococcus aureus, and we're going to talk about that this semester. Moderate numbers of normal microbiota are found in the genitals, the urethra, which is the tube from the bladder to outside. Your eyes, your stomach can have some normal microbiota. And little or no normal microbiota should be in the blood, the organs, and the internal body cavities. In fact, this is why when you get appendicitis and you're worrying about the appendix bursting, there's no normal microbiota in the body cavity, but if the intestines burst and all this bacteria from the intestine, if the appendix bursts and all this bacteria from the intestines floods into that area, this is a place where there's no bacteria normally, and it can be very dangerous. And I write little or no bacteria in there, little or no normal microbiota, because there are, again, with our techniques to look at the microbiome, microbial DNA has been detected in the brains, the lungs, the bladder, and some other parts of the body, but very few, if any, actual microorganisms have been found. So these ideas might be developing, but it's definitely clear that the number of microbes in our organs, in our blood, etc., are very low compared to other parts of the body. Why do we want to have normal microbiota? What are they doing for us? Well, there's all sorts of things, and it gets into a lot of details fast, but in kind of just some general things. They do help us digest some carbohydrates. You might notice that sometimes you get flatulence when you eat certain foods, and that might be because some carbohydrates that we don't digest, your microbes do, like some carbohydrates in beans and things. And so that gas is just a byproduct of microbial metabolism. Some of our microbes synthesize vitamins that we need, vitamin B12 and vitamin K. And a big one is what we call microbial antagonism. It says it right here, competing with, antagonizing, and neutralizing pathogens. And so this would be the concept of microbial antagonism. And so that idea is, I like to put this picture of a nice healthy lawn versus a lawn that has a lot of patches in it. And you can imagine if a weed seeds kind of floated along and landed in this nice healthy lawn, it wouldn't grow very well. Whereas if it landed on this kind of dirt area, you might have a chance of growing. And the idea is if your normal microbiota are like this healthy lawn, they're covering your surfaces, they're going to outcompete any pathogens, they're going to outcompete, they're going to be taking up space, they're going to be taking up nutrients, and there's just not going to be space for new microbes to grow. So transients that might be pathogens won't be able to grow in a well-populated area with normal microbiota. Whereas if maybe you took some antibiotics and your normal microbiota aren't as populated, you might be more likely to be harmed by a pathogen because there's going to be less competition for that pathogen and they might be able to grow. And we're going to talk about that kind of thing this semester. So microbial antagonism, normal microbiota discouraging the growth of transients and pathogens. And I think the last thing I have here is describe the characteristics of a healthy microbiome. And I just wanted to show you a couple things. If you look at these pictures, what they're showing you is they did some microbiome research and sampled all these different parts of the body. And all these colors represent different groups of bacteria. So here's the group names. And what you're noticing, a healthy microbiome is going to be different in different parts of the body. So if you look at these different regions, oh, here's the underarm, here's the cubital, antecubital region, the forearm, different places, you're going to have different mixtures of microbiota in the nose versus the arm based on these colors. And, the other thing we notice is even on a single person, you'll have different microbiota on your two different hands. The mixture can be a little bit different. So the first characteristic of a healthy microbiome is there's just there's different environments in the body, and so there's different microorganisms. We have all sorts of different ones throughout our body. This is another diagram that's kind of cool. HV means healthy volunteer. So here's 4 different people, healthy volunteer, 1, 2, 3, 4, and here they are again. And what they did is they sampled the antecubital fossa, that bend in your arm, in your elbow. Where am I? A little bend in your elbow. And what you can see in 4 different people, they found a different mixture. So even healthy people can have different microbiota from each other. So it can vary. And notice that these 4 different people were different from each other, but also their different locations in their bodies were different. So it's normal and healthy to have a variety of microorganisms, even from person to person. And then this last picture is showing you, again, here's the different colors. I'm going to go at this part on the top. The different colors represent different groups of microorganisms. And here's a single infant that they tested who was 3 days old, all the way up to 838 days old, like 3 years, almost 3 years old. And they tested this baby over a period of time. And what you can see is the microbiome got more complex as the baby got older. And different things happened. They introduced formula, and here the microbiome changed. They could have some changes based on taking antibiotics. And so the microbiome seemed to get more complex as the baby got older. And so if you look at that kind of in a general thing, we have 2 things listed here. Richness, which means variety, and stability, how much does that change? And so what they're showing you in infancy, they don't have a lot of variety. They don't have a lot of richness. And they're relatively unstable. The microbiome is changing a lot over short periods of time. As you get older, what you see with the richness, the variety, that by adolescence, you've got a lot of variety. And that stays the same as you go into late senior. The stability, the amount that the microbes change, again, that progresses as you go into adulthood, and the stability into adulthood is is very high. So by the time you're an adult, you have a lot of variety of microorganisms, and they don't change a lot. And this is considered what we know now about a healthy microbiome, that you've got a lot of them, and you've got a lot of variety, and they don't change a lot. And so I would call that your hallmark of a healthy microbiome. Now, what you see here as you get older is older people go backwards in this stability, their microbiomes start changing again. And it could be due to medication, menopause, illness, all sorts of different things can happen as you get older. And your microbiome is going to be destabilized, and it's going to contribute to overall, declining health. So a healthy microbiome's going to have a high richness, lots of different kinds of microorganisms, and they're going to be very stable. They're not going to change a lot. And I'm going to stop right there.